Hi, my dear student. Welcome to my channel. Today, discussing the topic 11th Standard Botany Living World Part 2. The topic viruses. Did you go through the headlines of newspaper in recent times? Have you heard of the terms Ebola, Zika, AIDS, SARS, H1N1, etc. There are serious entities which are considered as biological bacilli, can cause disease in man. They are called viruses. We have learned about the attributes of living world in the previous session. Now we shall discuss about the viruses which connect the living and non-living world. The word virus is derived from Latin meaning poison. Viruses are submicroscopic, obligate intracellular parasites. They have nucleic acid core surrounded by protein coat. Viruses in the native state contain only a single type of nucleic acid, which may be either DNA or RNA. The study of viruses is called virology. W. M. Stanley, in the year 1904 to 1971, an American scientist obtained crystalline protein sediment from infected tobacco juice in the year 1935. He was jointly awarded Nobel Prize with Dr. J. H. Northrop for chemistry in 1946. Next one, milestones in virology. 1796, Edward Jenner used vaccination for smallpox. 1886, Adolf Meyer demonstrated the infectious nature of tobacco mosaic virus using sap of mosaic leaves. In the year 1892, Dmitry Ivanovsky proved that viruses are smaller than bacteria. 1898, M. W. Pickering defined the infectious agent in tobacco leaves as Contagium vivum bludium. In the year 1915, F. W. Todd identified viral infection in bacteria. 1970, D. Hurley coined the term bacteriophage. 1984, Luc Montagnier and Robert Calo discovered HIV, full form Human Immunodeficiency Virus. Size and shape of virus. Viruses are ultra microscopic particles. They are smaller than bacteria and their diameter range from 20 to 300 nanometer. 1 nanometer is equal to 10 power minus 9 meters. Bacteriophage measures about 10 to 100 nanometer in size. The size of the TMV is 300 into 20 nanometer. Generally, viruses are three types of based on shape and symmetry. Number 1, cuboid symmetry, example adenovirus, herpes virus. Number 2, helical symmetry, example influenza virus, TMV. Third one, complex or atypical virus, example bacteriophage and vacina virus. Another one, characteristic features of viruses, living characters and non-living characters. Living characters, presence of nucleic acid and protein, capable of mutation, ability to multiply within living cell, able to infect and cause disease in living beings, show irritability, host specific. Non-living characters can be crystallized, absence of metabolism, inactive outside the host, do not show functional autonomy, energy producing enzyme system is absent. Another one, classification of viruses. Among various classifications proposed for viruses, the classification given by David Baltimore in the year 1971. The classification is based on the mechanism of RNA production The nature of the genome, that is single strand SS, double strand DS. RNA or DNA, the use of reverse transcriptase, mentioning by capital RT, and ssRNA may be plus sense or minus antisense. Viruses are classified into 7 classes. Class number 1, viruses with double stranded DNA, example adenoviruses. Class 2, viruses with sense single stranded DNA, example parvoviruses. Class 3, viruses with double stranded RNA, example reoviruses. Class 4, viruses with sense single stranded RNA, example togoviruses. Class 5, viruses with sense single stranded RNA, example rhabdoviruses. Class 6, viruses with sense single stranded RNA, reverse transcriptase that replicate with DNA intermediate in life cycle, example retroviruses. Class 7, viruses with double stranded DNA, reverse transcriptase that replicate with RNA intermediate in life cycle, example virus. Viral genome. Each virus possesses only one type of nucleic acid, either DNA or RNA. The nucleic acid may be a linear or circular form. Generally, the nucleic acid is present as a single unit, but in wound tumor virus and influenza virus, it is found in segments. The viruses possessing DNA are called deoxyviruses, whereas those possessing RNA are called riboviruses. Majority of animal and bacterial viruses are DNA virus. HIV is the animal virus which possesses RNA. Plant viruses generally contain RNA. Cauliflower mosaic virus possesses DNA. The nucleic acid may be single stranded or double stranded. On the basis of nature of nucleic acid, viruses are classified into 4 categories. They are viruses with single stranded DNA, parvoviruses; double stranded DNA, bacteriophages; single stranded RNA, tobacco mosaic viruses; and double stranded RNA, wound tumor virus. Another one, tobacco mosaic viruses. Tobacco mosaic virus was discovered in 1892 by Dmitry Ivanovsky from the tobacco plant. Viruses infect healthy plant through vectors like aphids, locusts, etc. The best visible symptom of TMB is discoloration of leaf color along the veins and show typical yellow and green mottling, which is the mosaic symptom. The downward curling and distortion of young apical leaves occurs, plant becomes stunted and yield is affected. Structure of TMV. Electron microscopy studies that revealed that TMV is a rod shape. Helical virus measuring about 280 into 150 micrometer with a molecular weight of 39 into 10 power 6 daltons. The virion is made up of 2 constituents, the protein coat called capsid and the core called nucleic acid. The protein coat is made up of approximately 2130 identical proteins of unit called capsomeres, which are present around a central single-stranded RNA molecule. The genetic information necessary for the formation of a complete TMV particle is contained in its RNA. The RNA consists of 6500 nucleotides. Another one, bacteriophage. Viruses infecting bacteria are called bacteriophages. It literally means eaters of bacteria. Phages are abundant in soil, sewage water, fruits, vegetables, and milk. Structure of T4 bacteriophage. The T4 bacteriophage tadpole shape and consists of head, collar, tail, base plate, and fibers. The head is hexagonal, which consists of about 2000 identical proteins of unit. The long helical tail consists of inner tubular core, which is connected to a head by collar. There is a base plate attached to the end of tail. The base plate contains 6 spikes and tail fibers. These fibers used to attach the page on the cell wall of bacterial host during replication. A double-stranded DNA molecule about 50 micrometer is tightly packed inside the head. The DNA is about 1000 times longer than page itself. Multiplication or life cycle of pages. Pages multiply through two different types of life cycle. One lytic or virulent cycle, second one lysogenic or avirulent life cycle. Number one, lytic cycle. During lytic cycle of page, disintegration of host bacterial cell occurs and the progeny virions are released. The steps involved in the lytic cycle are as follows. 1. Adsorption. Page T4 particle interact with the cell wall of host. The page tail makes contact between the two and tail fibers recognize the specific receptor site present on bacterial cell surface. The lipopolysaccharides of tail fibers act as receptor in pages. The process involving the recognition of page to bacterium is called landing. Once the contact is established between tail fibers and bacterial cell, tail fibers bend to anchor the pin and base plate to the cell surface. This step is called pinning. Second one, penetration. The penetration process involves mechanical and enzymatic digestion of the cell wall of the host. At the recognition site, page digests certain cell wall structure by viral enzyme that is called lysozyme. After pinning, the tail sheath contract and appears shorter and thicker. After contraction of the base plate enlarges through which DNA is injected into the cell wall without using metabolic energy. The steps involving injection of DNA particle alone into the bacterial cell is called transfection. The empty protein coat leaving outside the cell is known as host. Third one, synthesis. These steps involve the dehydration of bacterial chromosome, protein synthesis and DNA replication. The page nucleic acid takes over the host biosynthetic machinery. Host DNA gets inactivated and breaks down. Page DNA suppresses the synthesis of bacterial protein and directs the metabolism of the cell to synthesize the protein of the page particle, and simultaneously replication of page DNA also take place. Fourth one, assembly and maturation. The DNA of the page and protein coat are synthesized separately and are assembled to form page particle. The process of assembly, the page particle is known as maturation. After 20 minutes of infection, about 300 new pages are assembled. Fifth step, release. The page particle gets accumulated inside the host cell and are released by the lysis of host cell wall. Second one, lysogeny cycle. In the lysogeny cycle, the page DNA gets integrated into host DNA and gets multiplied along with nucleic acid of the host. No independent viral particle is formed. As soon as the page injects the linear DNA into the host cell, it becomes circular and integrates into the bacterial chromosome by recombination. The indicator page DNA now is called propage. The activity of the propage gene is repressed by two repressor proteins which are synthesized by page genes. This checks the synthesis of new pages within the host cell. However, each time the bacterial cell divide, the propage multiply along with the bacterial chromosome. On the exposure of UV radiation and chemical, the excision of page DNA may occur and result in lytic cycle. Virion. Virion is an intact infective virus particle which is non-replicating outside a host cell. Virioid. Virioid is a circular molecule of single-stranded RNA without a capsid and was discovered by T.O. Diner in the year 1971. The RNA of viriard has low molecular weight. Viriard cause citrus exocartis and potato spindle tuber disease in plants. Virizoids were discovered by J. W. Randalls and co-workers in the year 1981. They are smaller circular RNAs which are similar to virions, but they are always linked with larger molecules of the viral RNA. Next one, prions. Prions were discovered by Stanley B. Brusner in the year 1982, and the proteinaceous infectious particle. They are causative agent of about a dozen fatal degenerative disorders of the central nervous system of humans and other animals. For example, Creutzfeldt-Jakob disease (CJD), bovine spongiform encephalopathy, commonly known as mad cow disease, and scrapie disease of sheep. Another one, cyanopages. Viruses infecting blue-green algae are called cyanopages and are best reported by Safferman and Morris in the year 1963. Example, LPP1, Lympia plectonema pormidium. Similarly, Hollings reported virus infected cultivator mushroom and causing dieback disease. That is called mycoviruses or mycopages. The virus attacking fungi are called mycovirus or mycopages. Holing reported virus infecting cultivator mushroom and causing dieback diseases. Viral diseases. Viruses are known to cause disease in plants, animals, and human beings. Number one, plant diseases. Tobacco mosaic disease, cauliflower mosaic disease, sugarcane mosaic disease, potato leaf roll, Banjaitapa banana, leaf curl papaya, vein clearing of lady's finger, rice tungro disease, cucumber mosaic, tomato mosaic diseases. Another one, animal diseases, foot and mouth disease of cattle, rabies of dog, encephalomyelitis of horse. Another one, human diseases, common cold, hepatitis B, cancer, SARS, severe acute respiratory syndrome, AIDS, acquired immunodeficiency syndrome, rabies, mumps, polio, chikungunya, smallpox, chickenpox, measles. Another one, do you know box? The streaks of tulip flower are due to tulip breaking virus, which belong to Botany viridae group. Viruses are Buffalo viridae group are commercially exploited as insecticide. Cytoplasmic polyheterosis granulovirus and entomobox viruses are employed as potential insecticides. Evaluation. Question number 1. Which one of the following statement about virus is correct? Option A, possess their own metabolic system. Option B, they are facultative parasite. Option C, they contain DNA or RNA. Option D, enzymes are present. So the correct answer, option C, they contain DNA or RNA. Question number 2. Who had defined the infectious agent in tobacco leaves as Contagium vivum bludium? Option A, Dmitry Ivanovsky. Option B, M.W. Pickering. Option C, F.W. Todt. Option D, Edward Jenner. So the correct answer, option B, M.W. Pickering. Question number 3. Identify the criteria not used for classification of viruses. Option A, SS single stranded or double stranded. Option B, use of RT, reverse transcriptase. Option C, sense RNA or antisense RNA. Option D, reproduction. So the correct answer option D, reproduction. Question number 4, approximate number of capsomeres in TMV. Option A, 3120. Option B, 1203. Option C, 2130. Option D, 3021. So the correct answer option C, 2130. Question number 5, the indicator page DNA is called. Option A, propage. Option B, bacteriopage. Option C, cyanopage. Option D, mycopage. So the correct answer option A, propage. Question number 6, a virus with a double-stranded DNA is called as. Option A, parvoviruses. Option B, toga viruses. Option C, adenoviruses. Option D, retroviruses. So the correct answer option C, adenoviruses. Question number 7, vaccination for smallpox was discovered by. Option A, D. Curley. Option B, Edward Jenner. Option C, Robert Calo. Option D, F.W. Todd. So the correct answer option B, Edward Jenner. Question number 8, 1 nanometer is equal to. Option A, 10 power 9 meter. Option B, 10 power minus 9 meters. Option C, 10 power 8 meters. Option D, 10 power minus 6 meters. So the correct answer option B, 10 power minus 9 meters. Question number 9, size of TMV is equal to. Option A, 300 into 200 nanometer. Option B, 30 into 20 nanometer. Option C, 300 into 20 nanometer. Option D, 280 into 40 nanometer. So the correct answer option C, 300 into 20 nanometer. Question number 10, viruses were classified into 7 classes by. Option A, David Baltimore. Option B, Todd. Option C, Ehrenberg. Option D, Alexopoulos. So the correct answer option A, David Baltimore. Question number 11, mad cow disease is caused by. Option A, prions. Option B, virion. Option C, viroid. Option D, page. So the correct answer option A, prions. Question number 12, which of the following shows coiled RNA strand and capsomeres? Option A, poliovirus. Option B, tobacco mosaic virus. Option C, measles virus. Option D, retrovirus. So the correct answer option B, tobacco mosaic virus. Question number 13, virions differ from viruses in having. Option A, DNA molecule with a protein coat. Option B, DNA molecules without a protein coat. Option C, RNA molecule with a protein coat. Option D, RNA molecules without a protein coat. So the correct answer option D, RNA molecules without a protein coat. Question number 14, which of the following statements is wrong for virions? Option A, they lack a protein coat. Option B, they are smaller than viruses. Option C, they cause infections. Option D, their RNA is a high molecular weight. So the correct answer option D, their RNA is a high molecular weight. Question number 15, what are prions? Name one human disease and cattle disease caused by prions. Question number 16, define virion. Question number 17, how was the word virus derived? Question number 18, name the expression in viruses with regard to nucleic acid. Question number 19, what is propage? Question number 20, name 2 viral diseases affecting humans. Question number 21, how do virions differ from viruses? Question number 22, why are viruses considered to be a balanced bacil? Question number 23, what are 3 main symmetry of viruses? Question number 24, list some viral disease which occur in plants and humans. Question number 25, give a concise account on virulent cycle or explain lytic cycle of a page.